should like to welcome everyone to the first of the Front Row lectures commemorating this year, which is a centenary of science at Scripps Research. It is my great pleasure and honor indeed to introduce our president, Peter Schultz, to you. Pete is president and CEO of Scripps Research. He also holds the Sam Skaggs Presidential Chair. He has made many seminal contributions to both science and medicine, and these range across disciplines, from expanding the genetic code to the discovery of catalytic antibodies to the invention of molecular diversity technologies that have provided tools and therapeutics for problems across chemistry, biology, and medicine. The Caliber Skaggs Institute for Biomedical Research has really been his unique invention, bringing great science and drug discovery together in support of Scripps Research. He has founded 9 biotechnology companies and has co-authored more than 600 peer-reviewed publications. I can certainly say that as Pete's colleague for the past almost 22 years, this is only a part of who he is and who he continues to be for Scripps. So Pete, I'd like to thank you on behalf of our community of scientists and our community of support staff for your integrity, your commitment to building our institution, the scientific rigor that reflects your vision, the dreams, and the execution, in which almost everything that we have approached within the institution, either as individuals or collectively, has been enhanced by the impact of science and on patients that have benefited from your vision and its execution and your leadership. I should also like to thank you on behalf of the patients who day by day and unknowingly receive the treatments that have emerged from Scripps Research and its basic discoveries. They don't need to know where these discoveries came from, but we recognize that quietly, silently, the good that Scripps Research does spreads throughout the community and the world. And they really embody the essence of your vision for Scripps, which has been science changing life. And so with that, I should like to ask you to speak.
Thank you. Thanks a lot, Hugh. If you haven't figured it out yet, I pay Hugh's salary, okay? It's been 5 years since I gave, I think, the first Frontiers lecture, and a lot has happened in that 5 years. And I thought I would take the time to update you on what we've done, where we are, and where we're going. And as Hugh said, it's our 100th anniversary this year. Scripps was founded in 1924 with a gift from Ellen Browning Scripps. We were down at the Cove before moving up here. I wish we'd kept the real estate down there. But Scripps really expanded under the leadership of Frank Dixon and Richard Lerner to 6 departments today. And those leaders were responsible for really building the scientific excellence of Scripps, as reflected in Nobel Prizes awarded to Scripps scientists, Artem in 2021 and Barry in 2022. They also created a graduate program which is one of the leading graduate programs in the United States, both in chemistry and biology. And then more recently, over the last 5 or 6 years, we've expanded our mission from just doing basic research to really translating those scientific discoveries to new medicines and clinical insights that really directly impact patients. So that's kind of the history of where we are and where we've been. Nonetheless, we're probably, when people think of Scripps, they think of sharks and fish, okay? They don't think of science and biomedical research. So we're probably one of the best kept secrets, certainly in the public eye, not only at the national level, but also at the local level. Whereas, you know, among scientists, we're one of the most highly regarded institutions. And I thought I would take time to kind of correct that oversight today by doing something that I rarely, if ever, do, and that's to boast about Scripps and its accomplishments. So please bear with me. We're a small institution, only about 130 faculty, but we have an annual research budget of over $600 million. Over six Nobel Prizes have been awarded for work done at Scripps. We probably have more Wolf Prize winners than any other institution in the world. Nature ranks us among the top 5 independent research institutes in the world. We've contributed enormously to the San Diego industrial base, biotechnology base, with over 50 companies being formed from Scripps intellectual property. Just this week, one was acquired for $2 billion. 15 FDA-approved drugs and vaccines have come out of work that was done at Scripps. So we really have, in light of our very small size, a pretty extraordinary track record in science. But we're also involved in education. In 1989, Scripps formed a graduate program. And that graduate program has basically been in the top 10 since 1999, which is, again, really pretty remarkable given our small size. And the fact that we actually only have a fraction of the subdisciplines in biology and chemistry that most other institutions have. The graduate school is expanding. This year we had 12,000 applications. We actually brought in roughly 80 students. And importantly, we're on our way to being the first graduate school where every graduate student has an endowed fellowship, meaning that they can work for whomever they want on whatever science excites them most. So where next? That's where we are, that's where we've been. Where are we going? Well, scientific institutions and educational institutions have basically been the same for the last 200 years. And I think it's about time we started to ask whether what we've been doing for the last 200 years is what we should be doing for the next 200. So we're beginning to look at really what should be the model for a research institute in the next century. And Scripps is an ideal place to ask that question because Scripps really does push frontiers. It pushes the frontiers of science, but historically, it's also pushed the frontiers of institutions and overcoming institutional barriers to science. And so we're starting to look hard and think, how do we reinvent the Research Institute to expand the science we do, and importantly, the global impact of that science, the impact of that science on making new medicines, and the impact of those medicines on the public. And so, really, that boils down to 4 key parts of the vision. We want to continue to empower scientists to pursue really bold, high risk, high reward, transformative ideas. That's becoming harder nowadays with reduced federal funding of science. And the fact that federal funding of science is starting to focus more on science that has a high probability of success, but that's very incremental. And really what you want to do is push the frontiers. We also want to expand the mission of Scripps by figuring out ways to accelerate the translation of scientific discoveries to medicines that directly impact patients, okay? After all, the public are paying for our privilege of doing science, okay? And I think everybody who practices science, who has the privilege of practicing science, also we have an obligation to see the impact of that science to the public benefit. We want to create a new business model for scientific institutions that amplifies the investment in science. Great companies like Microsoft and Apple weren't built by doing IPOs, taking those monies, putting them in safe investments, and taking 5% of those investments and spending them every year. They were built by taking those revenues and reinvesting them in the companies to grow the companies and build the companies because they believed in the companies. And why don't research institutes do the same thing? And also, as we move, you know, what COVID did was make a lot of the world virtual, okay? And so why does a research institute have to be constrained by physical boundaries? You know, why do we have to be constrained by the land we own in La Jolla? Why can't you build an institute without walls that partners with other institutes to enable scientists collaboratively at other institutes to also do outstanding science in partnership with their institutions and Scripps? So that's really what we want to begin to build. But as we do that, we need to keep in mind that really the Scripps' core historical strength has been its outstanding science and technology development. Everything from understanding basic biology like the sense of touch, all the way to building new technologies that accelerate the way we make new medicines. And let me just take you through some of what we've done in the world of science. Now let's start with chemistry, probably the course everybody hated most in college if you were so inclined. But what chemistry does best is control the structure of matter at the molecular level, okay? And it's the only science that really does that. And I would argue that Scripps' chemistry department leads the world in that process, in chemical synthesis and in chemical biology. You know, great discoveries have been made that make it easier and more efficient to make molecules, asymmetric catalysis, CH activation, and others. Barry Sharpless invented click chemistry to make molecules very quickly and easily, which garnered the 2022 Nobel Prize in Chemistry. Methods to make molecules millions at a time were invented at Scripps, and methods to screen those molecules for new drugs. New chemical and proteomic tools to identify new targets to create new medicines in cancer and CNS disease. And harnessing the complex biology of the cell to make complex molecules and assemblies of molecules that are hard to make any other way. And also inventing drugs like tafamidis. It's the only medicine right now approved for cardiomyopathy. And that's chemistry. We also have a neuroscience department, and I would argue it is the leading center in the US for sensory neurobiology, understanding how the brain communicates not only with all the other organs in the body, but also the external world. And again, that was underscored by Artem winning the Nobel Prize for discovering how we sense touch. This department has also uncovered mechanisms of neurodegenerative disease, developmental diseases like autism, and the new faculty we're hiring are building very powerful new genomic and imaging tools to really understand how the brain communicates with the rest of the nervous system and the rest of the body. Scripps is also a leader in structural biology. Some of the most important structures in the living cell have been solved at Scripps. The structures of GPCRs that are membrane-bound proteins that transduce signals from outside the cell to the inside of the cell. Those structures were solved here. Key structures in understanding how the immune system recognizes foreign pathogens, both at the cellular level and at the level of the antibody molecule. Those structures were solved here. The structure of poliovirus was solved here. And the earliest SARS-CoV-2 structures were solved here. And those structures actually played a key role in enabling the development of the vaccines that almost all of us are certainly taking. New form of proteins, intrinsically disordered proteins, were characterized here, which changed the way we think about protein structure and structural genomics. We are the leading center in structural genomics, which is solving structures in a very high throughput, efficient mode to understand how the cell works. In molecular medicine, surfactant was invented here. You know, this is an inhaled drug that's given to infants that have infant respiratory distress syndrome. And if you've ever seen the videos of these purple infants actually are given an inhalation of this drug, and all of a sudden they're pink again, okay? It's pretty amazing. Hugh and Ed Roberts invented Saposia, which is a drug that was just approved for relapsing forms of multiple sclerosis, a new strategy for dealing with this debilitating disease. Patients who have hairy cell leukemia go into complete remission when they take lustatin, which was invented here. Understanding how the biological clock and circadian rhythm is regulated and its role in disease. Key discoveries were made at Scripps. Understanding how carbohydrates and the role carbohydrate and carbohydrate receptors play in cancer was pioneered at Scripps. And most recently, understanding the molecular basis of addiction and ways to actually treat addiction, not only on a daily basis, but for longer periods of time, were invented here. Immunology is also historically one of Scripps's major strengths. Scripps is the leading center for the development of broadly neutralizing vaccines for viruses, specifically HIV and influenza. It's been a pioneer in the study of viral pathogenesis and immunity to understanding immunity to viruses. Innate immunity was really first characterized at Scripps. Combinatorial antibody technology was invented at Scripps, which allows us to make antibody therapeutics very, very efficiently and quickly. New ways to identify, monitor, and treat pandemic viruses. We're one of the leading centers that's undertaken that mission. And finally, some of the accomplishments that we've had. But despite those accomplishments, okay, not only at Scripps, but around the world, I mean, our understanding of human biology over the last 30 years has just exploded. But still I ask, why are there so many unmet medical needs? Why do kids who have type 1 diabetes still take insulin, okay? Why, if you have pancreatic cancer, is your life expectancy so short? What about other viral pandemics, okay? What about all of the diseases, degenerative diseases of aging? There's a huge unmet medical need for this. And the question is, why is science going so quickly, and why is the development of new medicines going slowly? And it's because there are intrinsic barriers between the two. Most scientific discoveries are made in research institutes, nonprofit research institutes like Scripps, who in general don't have the tools to turn those discoveries into drugs. So those discoveries are turned into drugs by biotech and pharma, the biotech and pharma industry. And institutions like ours in pharma have very different cultures, very different processes, and at times very different motivations, which creates barriers, which really slows the translation of great science into great new medicines. And how do we deal with that? So people have tried and explored various models to accelerate that process, none of which have been terribly successful. So the question really remains is how do we best bridge scientific discoveries at the bench with drug discovery to accelerate the translation of those discoveries to medicines that directly impact you? And our answer was really to ask whether at Scripps, not only could we do great science, and can we do great science, but can we augment that great science with translational capabilities in two forms, in the form of Caliber SCAGs and the form of the Scripps Research Translational Institute, to use those centers to really seamlessly turn discoveries made at Scripps into new medicines, into new clinical insights and clinical advances that really impact human health. And so let me tell you about what we've done in those two areas over the last 5 years and the advances we've made really to augment the mission of Scripps. And first, let me talk about Caliber SCAGs. This was formed as an independent 501c3 nonprofit institute really to focus on making medicines, okay? And really bridging this valley of death that is this huge void between an idea and a drug that you actually put into patients and show efficacy. And what we did was we actually brought Caliber SCAGs Institute into Scripps as a division in 2018 to create what I think is a one-of-a-kind translational research center in the nonprofit world. It collaborates with foundations, with pharma, with other academic institutions all over the world. It has huge capabilities, technology platforms. It's actually put, in the short time it existed, 9 drugs into clinical trials in human patients and 5 more in IND-enabling studies, which is pretty much unprecedented by any nonprofit institute in the world. It operates completely autonomously. It makes its own decisions about what to work on and how to execute those programs and how to move them forward into patients. It owns all its intellectual properties, and it's developed quite strong financial stability to support our preclinical research efforts. It has state-of-the-art infrastructure. We've actually built out most of the capabilities, if not all, that biotech and pharma have, but in a nonprofit setting. And it works across a large range of disease modalities and cell therapies and the development of biologics to treat disease and in the development of small molecule drugs, all the way from protein engineering and small molecule discovery to IND-enabling studies and clinical trials. And surprisingly, it works across a huge number of unmet medical needs. It works on the 3 leading killers in the world, which are cancer, respiratory disease, and heart disease. But it also works on global diseases that impact low and middle income countries, metabolic, cardiovascular disease, and degenerative diseases of aging. And it works on problems that may be viewed too high risk or too early for the biotech and pharma industry to undertake. For example, how do we actually repair damage to the body and simply not just slow it down? And let me give you a few examples to give you ideas of what's been done over the past 5 years. One area that Caliber has undertaken are creating safe, effective cell therapies to treat cancer. Some of you have probably heard of CAR T cells. These are cells, they're immune cells that you take out of the body, engineer in a specific way to target cancer cells and put back into the body. And in lymphomas and certain leukemias, this approach, this treatment, has been basically curative, which is remarkable. But what you'd really like to do is now apply that to solid tumors like breast cancer, prostate cancer, colorectal cancer. And how do you do that? Well, the challenge here is you don't control cells like you control small molecules. If I take Advil until my headache's gone, and then I quit taking it, okay? If you take a cell therapy, the cells do what they want to do, okay? So they expand. It's hard to turn them off. It's hard to turn them off. It's hard to dose titrate them, which creates a safety problem that's really prevented this remarkable therapy from being applied to a larger range of cancers. So the folks at Caliber invented a technology to control these CAR T cells by making a switch. It's more like a light dimmer. And it allows you, a two-step process allows you to turn on the CAR T cell, increase the CAR T cell dose, and decrease the CAR T cell dose. And so these were all things we need to do to move this treatment into solid tumors. And so we carried out a phase 1 clinical trial, which surprisingly, maybe not, these CAR T cells in patients, lymphoma and leukemia patients, worked exactly as they were designed. They had very high responses, they had improved safety, they had improved engraftment, they lasted for a long time. And based on those results, we're now positioned, and we are moving this therapeutic modality into breast cancer in the first half of 2024. And if it does for breast cancer or prostate cancer what it's done for blood cancers, it'll really have a remarkable impact in the oncology space. Now, another area we've pursued, like I said, is regenerative medicine. Most drugs that we take simply slow the progression of disease. Okay, what you'd really like to do is stop the disease, and if you could, reverse the disease process and repair the damage done by the disease. And so we've undertaken a whole series of programs in regenerative medicine where we make drug-like molecules that work on endogenous stem cells in our body that make our organs, okay? We're not giving stem cell therapy, we're just giving drugs that work on endogenous stem cells. And one area we've looked at is respiratory disease. This is one of the leading killers in the world. And asked whether we could treat diseases like, idiopathic pulmonary fibrosis or COPD or other lung diseases with a drug that you inhale directly into the lung, expands lung stem cells, and basically makes new lung from the damaged lung, okay? And so this drug was discovered in a basic research, this strategy and drug were discovered in a basic research lab at Scripps by an assistant professor. And it turns out it was developed at Caliber to be an inhaled treatment. It's safe. It works in every respiratory disease model we've put it into, okay? It induces selectively expansion of lung stem cells. It works in combination with other standards of care. And so we're on track to initiate clinical trials with this drug in patients at a site in Europe in the first half of 2024, which would be a first-in-class therapy to repair a lung. Can you do the same thing with heart disease? I actually have to see a cardiologist, okay? The good thing if you're running something is you can suggest to people what they work on, okay? So we've actually worked on heart repair too. What happens if you have heart failure, okay? Nowadays you don't die, okay? In the old days you died very quickly. But you have irreversible damage to the heart and your heart progressively loses function, the quality of your life goes down, and you eventually die. What if after a heart attack, you could give a drug that would repair the heart and make new heart and prevent scarring and improve heart function? So there's a pathway that's been shown if you activate it genetically, you can do exactly that, the YAP pathway. And it's been shown in both rodents and in pigs. The problem is it's really hard to turn the YAP pathway on genetically in the heart. So we invented, again, out of a basic research labs at Scripps, came a drug that selectively expands heart cells, cardiomyocytes, okay? And you can pericardially administer this to the heart selectively, okay? It has very low systemic exposure. And what it does in rodents is it actually increases heart function. It increases ejection fraction pretty dramatically, okay? It makes new heart muscle, and it limits scarring, okay? And we've now shown in PK/PD studies that it seems to work the same way in pigs, which is the best model for human heart disease. And so we're now moving this into the pig efficacy models, and our hope is if it works, and we're working with some of the best cardiologists in the United States, this is really going to transform the treatment of heart failure, okay? It's going to repair the damage, again, for the first time. How about intestinal bowel disease? Many of us suffer from Crohn's or ulcerative colitis. And, you know, the treatment for that is to suppress inflammation, okay? So you take anti-inflammatories the rest of your life. What you really want to do is repair the barrier between the microbiome in your gut and the rest of your body so you don't get leakage of bacteria and you don't get this inflammation. So how do you repair that barrier? Well, there's a natural peptide called GLP-2 that's responsible for maintaining that intestinal barrier. And we've used the technology platforms to engineer a very potent, highly stable form of that molecule with a long half-life. And we put that into human trials. It appears to be perfectly safe, has a very long half-life, and it upregulates biomarkers that suggest that it'll be very efficacious. So we're partnering this with AbbVie because they have Humira as the anti-inflammatory, and now they want to repair damage in addition to suppressing the inflammation. And this could change the way we treat IBD. How about neurodegenerative disease? There's this huge realization that the immune system plays a really important role in neurodegeneration, and neuroinflammation is a key driver. And so if you can suppress neuroinflammation, you can treat a variety of neurodegenerative diseases like Alzheimer's, Parkinson's, ALS. And it's been shown in investigator-led trials that upregulating Treg cells, cells that regulate the immune response, is actually beneficial to all three of those classes of patients. The problem is the way we can do that today uses a protein that's very short-lived and very immunogenic. So again, we use the technology platform at Caliber to make an antibody fusion of this factor. And with Michael J. Fox Foundation and ADDF, we've advanced this now into patients. And again, it looks to be very safe And it actually it downregulates cytokines that we think show that it will likely have the effect we hope it to have in a variety of neurodegenerative diseases. We've all heard all the time about anti-obesity drugs, okay? And why are we interested in anti-obesity drugs? I mean, some of us are interested in anti-obesity drugs because we want to look thinner, okay? But obesity is really a driver for a huge number of obesity-related diseases, for diabetes, for heart and liver disease, for osteoarthritis, and even for neurodegenerative diseases. So if you combat obesity, it's likely to have a huge health impact around the world. And probably the best drug to combat obesity is made by Lilly. It's tirzepatide, Mounjaro. And it acts on two what are called incretin receptors. And by acting on two receptors instead of one, it has a very, very impressive effect on weight loss and glucose homeostasis. The problem is it's an injectable. And most of us who need to lose 20 or 30 pounds don't want to take an injectable all the time. What we want is an oral pill we take once a day or once a week. And if you had that, I think there would be a huge compliance, and you would reduce a lot of these comorbidities. So we've actually used technology again to make a stable, highly potent, oral dual agonist that it looks like you can take once a day as a pill. And now we're working on one that we believe you could take once a week as a pill. And once you do that, I think many, many people will actually begin to treat their own obesity and avoid some of these other health issues. Again, this is right now in IND-enabling studies, and we hope to advance it into human trials next year. We also work on diseases that affect low and middle income countries where there isn't a really strong commercial driver to work on these. One is HIV. You know, we think about the number of people COVID killed in the last few years. HIV has killed over 500,000 people a year for 40 years, okay? It's a huge, huge problem. And we still, despite all the work being done around the world and at Scripps, we still don't have a vaccine. But what people are starting to show is you can make long-acting small molecule drugs. And so with funding from the Gates Foundation, we've actually managed to make a drug, a small molecule drug, that could have up to a 6-month half-life in patients. So you go into your doctor once every 6 months and get a sub-Q injection. And if you have HIV, you're treated. And if you don't have it, you can't get it. So this could do away. If you give this to all the women in low and middle income countries, okay, you could probably do away with the spread of HIV. Gilead and Merck, who are two of the most productive pharmaceutical companies in the treatment of HIV and development of HIV medicines, has partnered with us. And human trials with this new medicine are ongoing right now. Tuberculosis. Tuberculosis kills more than 1 million people a year, okay? Primarily, again, in low and middle income countries. The current treatments for TB are 9 months. So what happens if you're taking a drug for 9 months? Well, after 3 or 4 months, you start to feel better. What do? You quit taking the drug because it's awful, okay? What happens is the TB comes back, but now it's drug resistant. Nothing kills it. So that's the problem with TB. So everyone's searching for drugs that shorten the treatment time and improve compliance. And we did that in collaboration with a group at Cornell in a very complex intramacrophage screen that I won't go into. But we found a drug that by itself really doesn't do much. It modulates cholesterol metabolism. But when you combine it with the standard of care, it's absolutely sterilizing in a very short period of time. So the Gates Foundation has gotten very excited about this and are making it one of their their highest priority programs, and they really want to put it into, proof of efficacy phase 3 trials by 2026. This could change the way we treat TB. The other big killer is malaria, okay? And people are really working on new anti-malarials because there's resistance to the existing drugs. What do you do? So what people would really like to do is find a drug where one pill cures malaria. Why is that? Well, if you give somebody in the developing world 3 pills and they take the first one, they feel better. What do they do with the other 2? They save it for their kids in case their kids get malaria, okay? If you have a 1-dose cure, you know they're going to be cured. Not only could that cure it, but it could prevent contracting malaria for the period of 6 months. So again, we managed to make a medicine that looks like it might do this. And again, that was developed early on with the Gates Foundation, but the John Martin Foundation is actually supporting the IND-enabling studies of this drug, hopefully, again, to go to patients later this year or next year. So those are a couple of examples of what we've done. And as you make those medicines, you feel good for 2 reasons. The most important reason is you're actually potentially impacting patients and saving lives, which is a great feeling. But the other feeling is, as you move these forward into the clinic and you show they're going to work, you partner them with pharma and you get revenues back, royalties, milestone payments, and so forth. And the nice thing there is actually those financial returns to the Institute allow us to begin to create a flywheel-like model that every time we succeed, we get rewarded, okay? And we can reinvest that reward in new drugs and new clinical trials and make new medicines. So the whole thing grows, okay? And so we're doing that. We're taking the revenues we've gotten from some of the medicines I described. We now have a STING agonist that we're moving into bladder cancer. About 100,000 people get bladder cancer a year. Half of them either can't take or BCG unresponsive. What do you do for those patients? Well, we think a STING agonist might be the best treatment for those patients and may actually be the best treatment for any patient. So we're actually now moving this through IND-enabling studies. We're talking with the best clinicians in the United States, and we're going to move this into patients using the revenues we got from creating those other drugs. On a slightly different note, we in-licensed an oral PD-L1 inhibitor. What are these? These are checkpoint inhibitors. So checkpoint inhibitors, you've heard of Keytruda, have probably been the biggest advance in cancer therapy in the last 30 or 40 years. But they're very expensive. They're biologics. They require infusions. They require biopsies. They require typing of tumors. And so more than half the world can't access the most important advance in cancer therapy in the last 30 years. What do you do? Well, if you had an oral, safe checkpoint inhibitor, okay, you could actually give that to patients in low and middle income countries. And in fact, you wouldn't even have to biopsy the tumor if it's a safe drug. You just give it to all comers who have, say, lung cancer. 1/3 of them are going to benefit. 2/3 may not, but it's safe. So you're going to save 1/3 of the patients or impact them pretty substantially. So this is a chance now with this kind of small molecule, low-cost, safe drug to move a major cancer therapy into the developing world. And we're now building a clinical plan and collaborations and understanding the cost to do this as well. And again, we're going to reinvest the revenues we've generated from those other medicines into this medicine. Now, there's probably no commercial value in doing this. There's a huge human value, okay, human life value. But, you know, if there happens to be a God and a St. Peter and a pearly gates, it'll be a hell of a story, okay? So anyway, so we're doing things like this, and we're also building a pipeline, because this is not build it, sell it, and start over again. This is building a renewable capability to turn scientific discoveries into new medicines. Now the other approach we're taking is with Scripps Research Translational Institute. This institute was founded by Eric in 2007 and merged with Scripps in 2017. And it has a slightly different focus. Its focus is on understanding human disease at the clinical level and at the level of the individual patient. Because right now we're making medicines and we test them in a huge number of people, 200 or 300 patients. And they may work great in 10, but not in the other 290, and they'll be killed, okay? So Eric's trying to understand disease at the level, and SRTI are trying to understand the disease at the level of individuals. And it's very likely diseases differ in how you want to treat them and how they progress in individual patients. And they do this using genomics tools, digital medicine technologies, AI, and other informatics technologies. And they also, the theme here is to bring together clinical investigators with basic scientists and with other disciplines to work together, okay, in a unique environment that you may not see in pharma. And so let me tell you about a couple of things that are going on in SRTI. The NIH put together probably one of the largest, if not certainly the largest public effort to create a huge database, okay, a health-related database of thousands and thousands of patients, okay, to begin to understand their diseases, their responses to medicines, and how their diseases progress at the level of the individual by accumulating a huge amount of information about each one of those individuals. Not only is that to create precision medicines that better serve individual patients, but it's also to decrease health disparities across the United States and across the world. And the NIH asked SRTI to lead this program, and SRTI is managing this program and managing a $250 million NIH grant, which is probably the largest NIH grant ever given for health-related science. And they must be doing a really great job because it was just renewed for $282 million, okay? So I think this initiative is likely to create whole new insight, not at the level of creating new medicines, but how do we use those medicines to better treat patients by better understanding patient outcomes. The other big effort that SRTI has undertaken is digital medicine, okay? Right now, when you do clinical trials and you look at patient responses, they're in the hospital. It's a very expensive, involved process. Why don't we use digital medicine, give people sensors and devices and surveys and look at electronic health records for them to take home and simplify that whole process and gather far more data about how they respond to individual medicine. So SRTI is leading this, and one of the big initiatives they have undertaken is Power Mom, which is really looking at over 6,000 pregnant women, okay, and understanding how their pregnancies vary across individuals, okay? And the idea is if you accumulate that database using a whole series of sensor devices and surveys and other electronic mechanisms, you'll really be able to begin to fill in knowledge gaps about who has the potential to get preeclampsia or gestational diabetes, or who's going to have peripartum depression. And then you can get ahead of those problems before they occur, okay? And so SRTI is leading this initiative as a consortium. And it's also a hub for what's called the Scripps Hub Academic Research Corps, SHARC, okay? But this is, again, an NIH-funded institute. And its goal is really to bring resources and expertise and partnerships to bear at both the national level, but really importantly at local levels to improve health and outcomes, patient outcomes. And it's really a collaborative effort across the large number of disciplines. It funds basic research, it funds clinicians, funds other investigators, and it's collaborations already are impacting San Diego very much, not only here at Scripps, but Scripps Health, Rady's Children Institute, and San Diego State. So this is an effort to bring together clinicians and scientists across all of San Diego to improve human health. And again, it must be successful because it was just renewed with a $50 million grant. So that's what we're doing to move the whole model for research institutes forward, okay? Not only to do great science and make discoveries and better understand, the physical and biological world around us, but to take those discoveries and accelerate the impact on the public, okay? Which I think we have an obligation to do. So as we move to the next 100 years, just to reiterate, Scripps has been kind of a risk-taking institution, and we're really focused now on taking more risk, but doing that to pioneer a new model for research and educational institutions that's very different than the last 200 years, okay? One that really removes the conventional boundaries that constrain what we do such that we can really expand our impact on science, medicine, and the public. And there are really three components. One is to continue to enable exceptional scientists, okay? We're hiring some of the best and brightest young scientists in the world, and we really want to give them the best tools in the world to pursue their science. We also are bringing in and training some of the best graduate students and postdocs in the world, okay? And ideally, we'd like to be the first institution in the United States that endows all of its graduate students with fellowships, much like Langone has done in the medical school. We want to accelerate the impact of that science on human health, and I've described what we've done in the past 5-8 years to do that. We've made really tremendous progress, but we have huge challenges ahead of us and opportunities ahead of us. We really do want to be the leader in the nonprofit world in translational research. And I think already we may be. And we really want to focus, like I've said, as we all get older, there are a huge number of age-related diseases. What are we going to do about them, okay? There's a huge opportunity in medicine to either stop or reverse those disease processes, okay? Increase quality of life, increase longevity. How about stopping pain without addiction? That's another huge opportunity that basic science is giving new insights into. Improve cancer survival, okay? What about the next major pandemic? If it's the flu, it's going to be very different than COVID. COVID affected the elderly, okay? It didn't really infect the young. Influenza is going to do both, okay? So there are going to be a lot of young people in the hospital and dying. And maybe we should start to think about that now rather than before it happens, okay? And how about diseases that affect low and middle income countries where you can't afford the best and most modern therapies? So this, again, represents a huge opportunity for us moving into the next 100 years. And finally, we want to think about a new way of doing science as an institution. Why should we be constrained by four walls in the real estate we have here? Why don't we not only enable scientists at Scripps, but also enable scientists at other institutions. It's collaborations between Scripps and the best scientists at MIT or Caltech or Stanford or wherever. Think about an institute of virtual faculty without walls, where you're just identifying the best scientists, enabling them to do their best science across not only human health, but also energy and the environment. So this is where we're headed in the next 100 years, and certainly we need your help and your partnership to accomplish all of these goals. We're committed to them. I'm 67, so the clock is ticking, okay? So we need to get this done sooner rather than later, and we very much need your help. And finally, I'd really like to thank you. Thank all the people that did all the wonderful things I talked about, but thank all the people in the community that supported all of those wonderful things. And without that support, we wouldn't have been able to do it. So thanks to all of you, and that's all I had to say. Go ahead. How much do we need from you? I hate to say it, but as much as you're willing to give. I mean, we're really, trying to do something audacious, okay? And, even a clinical trial today to test one of these medicines, it's $25 million, okay? And just to put a drug into patients and see if it works. And if it doesn't work, it's $25 million gone, okay? So there's a huge challenge there, and how do you fund those clinical trials? And you have to fund those clinical trials, okay, to make those new medicines, especially if pharma is too high risk or too crazy or not commercially viable for pharma. So even that, but as you think about doing science, okay, for a while Scripps had a fund. It was provided by the Skaggs family that gave roughly $400K unrestricted research funds to leading scientists at Scripps, okay? And what did that enable? It enabled probably half of what I talked about. The two Nobel laureates that I just described who got Nobel Prizes in 2021 and 2022, that research was funded by those unrestricted funds, not by NIH because it was viewed as high risk, okay? So providing funds that allow people to take risk and do things that transform our understanding of science is a huge need, okay? The investigator program, why fund just those people at Scripps? Why not pick the best people across the United States who are working on things we might not work on here, like new energy, environment, and whatever, and enable them as a collaboration with faculty at other major institutions? They would love that. And you expand the Scripps footprint. Why aren't we doing that? So there's this huge number of opportunities, okay, that we really are trying to tackle. And, we're creating our own resources for doing that by making drugs and benefiting from the commercial value we create, okay? But, that'll take a longer time to do it without your help.
Thank you very much. It is wonderful, all the new drugs that are coming out and helping humanity and curing diseases. I just want to address one other section. My son-in-law's wife has lupus and cancer, and they have a drug that they're taking at $2,800 a month. He's gone into bankruptcy, he's mortgaged his house, and doesn't know if he can keep going. We have these wonderful drugs, but there's some people that just can't afford them. How do we address this problem?
Yeah, so that's a problem that I think requires more than research institutes contributing to the solution of. It's also a problem at the political and governmental and commercial level. Medicines do take a lot to develop. They are expensive, and there are far more failures than there are successes. But That's not to say that some medicines should be far more affordable. And so one thing we've done, especially we started it in our collaboration with the Gates Foundation, because the Gates Foundation and us with them are focused on people who can't afford. They can't afford $1 a day for a drug, okay? You're talking about 10 cents to save a life, okay? So what we did is with the Gates Foundation, we created by far the largest collection of all known drugs that have been put into people, that are approved, that have been at late stage preclinical, and are asking whether we can repurpose those drugs. And if they're off patent, they're very inexpensive. By doing that, we found new drugs for cryptosporidium, okay, and other diseases that affect low and middle income countries. But we're now beginning to apply that collection of known drugs and the idea of drug repurposing to cancer drugs. We found a very interesting drug that's been used for malaria for a disease called myelodysplastic syndrome that leads to AML, for example. It's cheap. It's used to treat malaria. So if that works, then all of a sudden you have a very low-cost medicine to treat a disease for which there are no effective treatments right now. So that's one thing we're doing to actually help that problem, but that's a complex problem, and I agree. It's a real challenge when a life-saving drug costs $350,000, okay? It just doesn't make sense.
Well, along the same direction, looking at the ecosystem, is among, a couple of components like academic center, the community, the pharmaceutical and manufacturing. Sometimes when we find, a wonderful solution, for instance, like CAR T cell, therapy in treating of multiple myeloma and some other lymphoma, it's a wonderful discovery and wonderful finding, wonderful manufacturing, as earlier alluded to, more than 500,000 or even more. One thing we can do is to look into the ecosystem, look into the, biochemical manufacturing, not just the pure discovery science, but also from a,
I agree what you're saying is
perspective.
The cost of medicines is a complex problem that requires a lot of inputs to solve, okay? And I think we're working on some of those, but we need help. The manufacturing at low cost, the kinds of clinical trials SRTI is starting to design where maybe all of a sudden you lower the cost of clinical trials dramatically, okay? And improve the outcomes, or at least improve the outcomes of that medicine for a smaller fraction of patients. So there are many, many different things that I think could and should be done to address that problem.
I have a two-part question. First, this is an amazing amount of information to follow. So will this presentation be available online so I can go look at it and try and remember it? And secondly, how do we follow, say, certain things and how they are progressing? Like if the thing on the technology on TB, those drugs. If somebody wanted to follow what you talked about today, where would we get that information?
So those are very good questions. And again, I think historically we haven't paid much attention to communicating with the rest of the world. We've been too busy doing science, okay? But we're really working hard, and I think we're taking the occasion and maybe the excuse of our 100th anniversary to really begin to communicate to the rest of the world what we're doing. And this is a start. I'm sure, Chris, that this will be, Chris, is this going to be online? Yes. So this will be available. It's hard enough for me to give it, more or less for you to absorb it in a half hour, okay? But we're also, we're expanding our communications team. We're bringing in, experts from the world of medicine that are writers to the lay people to communicate these. So I would say you've identified one of the reasons that Scripps is thought of when people think about Scripps, they think about aquariums, okay? And 2024, we're really going to make a large effort to change that. And I've been asked that question before, and I think our communications group is really sensitized to that challenge. But it's a very good point.
And I think now, Pete, there is an opportunity for people to mix and speak to
Great.
you afterwards.
One last question.
Sure, go ahead.
You've done great, amazing work. And while we're on it, I mean, you've got great support from people like foundations like Gates. Who are the dream foundations and organizations, your dream foundations, that you would like to create a network and relationship to help carry this forward? Put those names of places that you know that are not playing with us. Please put them out for us.
Yeah, well, I think the great thing is, I think the Gates Foundation is kind of, blazed the trail for others to follow, not only other foundations to follow, like Wellcome Trust and others, okay, but also other philanthropists to follow with the Giving Pledge and so forth. I think, the Gates Foundation is very focused on infectious disease. And I talked with Bill about spending $10 million to bring cancer therapies to the developing world, and that's just not on their radar. So there must be other people that it is, okay? I think our biggest challenge in 2024 is actually creating connections with those people, because there are a huge number of foundations. We just formed a foundation with Helmsley Trust, okay? Also with ADDF, also with Michael J. Fox, okay? With CERN, okay? With others. But there's more than those foundations. There's a lot of philanthropists that really want to have an impact, okay? And they don't know how, because maybe they didn't make their money by making drugs, okay? Maybe they made their money in, IT and other areas, and they don't know how to do it. And making those connections, so A, they don't waste their money, and B, their money has the biggest possible impact. And not an impact of building the endowment and spending 5% a year, okay, to do something. Really actually doing something with their money that actually has an impact and actually amplifies.
And the trick is, who do we know?
And that is what we are also building a team to decide. And we are getting help. We have a really amazing board. We have great people like Ron Burkle and Gerald Chan and Herbie Wertheim and Gurley and others, Jean Lay and others on the board that are really interested in this problem and really understand the impact we can have and what we need to have that impact. And so they're beginning an outreach to the rest of their network to really begin to connect us. And we're looking for other ways to do that. But I think there is just, it's two ships passing in the night, and you need to somehow avoid that. So those two questions probably have identified the areas that I'm going to spend most of my time on in 2024.